Okay, well, welcome to today's Safe World by Design webinar. And today we're covering an important topic, which we're having increasing use of toxicogenomics, not only in mechanistic toxicology and research, but now it's looking increasingly promising. We can use it supporting practical risk assessment problems, which could be classification or points of departure for risk assessment. And so our topic today is very much in that area. How can we extract signatures, biomarkers from such data? And with a focus on genotoxicity assessment, which is a key important aspect of any compound risk assessment. And I'm very happy to have a younger emerging scientist from the Vrije Universiteit of Brussels, from the group there, who's working on her Ph.D. on this topic, and will be our presenter today. So Anouck Thienpont, and she will take you through this topic over the next 20 minutes or so, and then you'll all have a chance to ask your questions through the question box, and we'll have a discussion of the topic at the end. So with that, I'm going to leave you. Please go ahead and make your presentation, Anouck.
Thank you, Barry, for the nice introduction. So thank you for having me here. And today I would like to present to you our work of the GENOMARK biomarker in human HepaRG cells as a new approach methodology for genotoxicity assessment. So I will explain a little bit more about its background, the development of the biomarker, a case study where we used it to de-risk misleading positives, also a case study where we used it in a more quantitative assessment for potency ranking of genotoxicants, and also a study where we investigated how to improve or increase the throughput of the GENOMARK biomarker. But before that, I would like to raise the question, why do we need new in vitro genotoxicity tests? Well, the first reason is the low specificity of the current in vitro genotoxicity tests, which results in a high number of misleading positives. And misleading positives are chemicals that show a positive result in one or more of the in vitro assays, but finally show the negative result in the in vivo assays. And these misleading positives trigger, therefore, unnecessary animal assays, which are timely, cost-consuming, and not always biologically relevant to humans. A second limitation is the limited quantitative use of the in vitro data. So at this moment, the in vitro genotoxicity data is primarily used in a qualitative binary way for hazard identification. And a final limitation is a lack of integration of new approach methodologies. And one interesting domain of new approach methodologies are indeed assays that evaluate the changes in the gene expression profiles after exposure to a chemical of interest. And different technologies such as qPCR, microarrays, or RNA sequencing are able to identify such gene expression profiles or signatures. And by combining such gene expression profiles with machine learning algorithms, new predictive models for genotoxicity could be developed. And this is why in a previous PhD project in collaboration between the VUB and Sciensano, the GENOMARK biomarker was developed. So GENOMARK is seen as an 84 biomarker gene biomarker that is able to classify chemicals of interest into genotoxic or a non-genotoxic group. And these 84 biomarker genes were previously derived from microarray experiments. And therefore, human HepaRG cells were exposed to 24 reference chemicals, 12 known in vivo genotoxic chemicals having a broad range of genotoxic mechanisms, and 12 known in vivo non-genotoxic chemicals. So the human HepaRG cells were exposed for a duration of 72 hours to the low cytotoxic concentration, the IC10, of the 24 reference chemicals. So afterwards, the microarrays were performed, and the transcriptomic data obtained demonstrated a gene signature, or let's say a fingerprint that could be used for future predictions. So later on, The microarray was translated into an easy-to-handle qPCR assay, and therefore the high amount of probe sets that we obtained with the microarrays were subjected to 3 different statistical analyses to obtain the 84 genotoxic specific genes, which were easily to spot in a 96 well plate to perform the qPCR assay. So when performing the qPCR, then we obtain the gene expression data, and these gene expression data is then subjected to a machine learning algorithm based on support vector machine to predict whether a chemical is classified as genotoxic or non-genotoxic. However, the prediction model that was previously developed was still open for some improvements, and that's why we also developed new prediction models. So initially, the tool was developed to use as for hazard identification, since at that moment we only tested at 1 concentration, the IC10. So since I previously just mentioned, the previously prediction model was still open for some improvements, and that's why we developed a new prediction models for GENOMARK based on machine learning. We first compared unsupervised machine learning algorithms with supervised machine learning algorithms. However, the unsupervised machine learning algorithms were not sufficient to distinguish the genotoxic from non-genotoxic chemicals, and therefore we needed to move to supervised machine learning algorithms to develop the new prediction model. In a first step, we created an extended data set of 38 chemicals, equally balanced into 19 genotoxic and 19 non-genotoxic chemicals, also including the 24 reference chemicals as mentioned in the previous slide. We also included the gene expression data of 9 chemicals of the publication of Otis et al. And we generated new gene expression data by PCR for 5 chemicals. So in total, we had an extended data set of 38 chemicals, and 2 different supervised machine learning algorithms were applied on the gene expression data. So the first algorithm was support vector machine, and the second one was random forest to develop a new prediction model. Both prediction models give an output or prediction score for genotoxicity between 0 and 1. And when the chemical has a probability prediction score below 0.45, the chemical is classified as non-genotoxic. Having a probability prediction higher than 0.56, the chemical is classified as genotoxic. When having a probability prediction in between, or when both models have a different probability prediction, the chemical is classified as equivocal. However, to facilitate data analysis and to make GENOMARK available for people who have no background in software programming or coding, we made GENOMARK available in an online tool or web application that is freely accessible for everyone. So the development of the new prediction models are also in our recently published publication in Altex. So this is a screenshot of the interface of our online web application, and here researchers can upload their gene expression data as log2fold changes. You also can download an example input file to test a little bit with the tool. Then both prediction models are run behind the screens, and as an output, you get a heat map with the gene expression data, as well as the individual prediction probabilities for each of the prediction model as an overall classification result, which could be easily downloaded. So after the development of the new prediction models, we wanted to assess the prediction performance of GENOMARK to de-risk misleading positives. And therefore, we performed a case study with 10 misleading positives that we selected based on different databases, such as the recommended database by Kirkland et al., expert opinions based on the SCCS, and other scientific literature. So the 10 misleading positives that we included are listed in the table on the slide with its corresponding in vitro and in vivo genotoxicity historical data. And in the first step, we determined the low cytotoxic concentration by a cell viability assay, and after that, we performed the PCR. So both prediction models were applied on the gene expression data, and the probability predictions of the random forest model are depicted in the left column, and the probability predictions made by the support vector machine prediction model are in the right column. So 6 out of the 10 chemicals were clearly classified as non-genotoxic by both models. 1 chemical, glyceraldehyde, was classified as genotoxic by both models, and 3 of the 10 were classified differently by both models. So overall, we demonstrated that GENOMARK had a high prediction performance to de-risk misleading positives, and therefore, in the context of use, we proposed to use GENOMARK in the toolbox of genotoxicity testing. So for example, having a chemical ingredient of interest, this can be a drug or a cosmetic ingredient or a food additive, for example, we propose to use GENOMARK as a first rapid screening to have an idea about its genotoxic potential. Or for example, as a follow-up on the current in vitro 2 test battery, together with other NAMs to de-risk misleading positives in a weight of evidence approach. So as I mentioned in the beginning, one of the limitations of the current in vitro assays is the limited quantitative use of the data. And therefore, in this case study, we wanted to investigate whether we could use GENOMARK for potency ranking of genotoxicants. And in this case study, we used 2 known genotoxicants, and we used the benchmark dose approach as a quantitative analysis approach. So the benchmark dose is defined as a dose or exposure that corresponds to a predetermined % change, or the benchmark response, BMR, in an adverse effect compared to the response in the controls. And one of the advantages to use the benchmark dose approach as a quantitative methodology is that the benchmark dose comes together with a 90% confidence interval, which is a measure of uncertainty of the estimation of the benchmark dose. So we use the benchmark dose approach by using the PRO software, which was developed by the RIVM and which is also freely accessible to everyone. So the 2 known genotoxicants that we selected for this case study was, in one hand, ethyl methanesulfonate, which is a direct genotoxicant based on the DNA alkylation, and the second one was aflatoxin B1, which requires metabolization. And this is also the chemical that we wanted to select to assess our metabolic capacity of the human HepaRG cells. So as both chemicals already demonstrated that they were genotoxic at the low cytotoxic concentration by GENOMARK, we now selected different concentrations around the IC10, and human HepaRG cells were exposed for 72 hours in 3 different batches. As a first step before performing the benchmark dose, we did a pre-filtering of differentially expressed genes. So the gene expression data was subjected to the student t-test to derive the differentially expressed genes with a fold change higher than 1.5 and a p-value below 0.05. We did the same exercise to derive the differentially expressed genes having a fold change higher than 2 and a p-value below 0.05. And as a result, the differentially expressed genes for aflatoxin B1 having a fold change higher than 1.5 were 55 out of the 84 biomarker genes. And for EMS, we had 63 out of the 84 biomarker genes. However, to compare the potency, we only wanted to continue with the amount of differentially expressed genes that were common in both data sets. And therefore, we continued with 41 out of the 84 genes as differentially expressed, having a fold change higher than 1.5. And we did the same exercise for the genes having a fold change higher than 2. And here we received 34 out of the 84 genes. As the data settings improved, we used a covariate analysis since the compounds were compared as for their potency, and we used a benchmark response of 10% or 0.1. As a result of PROST, we got for each of the differentially expressed genes the estimated benchmark dose with its corresponding 90% confidence interval for aflatoxin B1 in orange and for EMS in blue. So here it's important to know that since the confidence intervals are giving an idea about the quality of the dose response data and also the uncertainty around the estimated benchmark dose, it's important that we can only compare the potencies when all the confidence intervals are clearly separated between the chemicals. And in this case, we see clearly that all the confidence intervals for aflatoxin B1 are clearly separated from the confidence intervals of EMS. So in this case study, as we see that aflatoxin B1 is more on the left side of the x-axis, which means an increasing potency, we can conclude that aflatoxin B1 has a higher potency than EMS in human HepaRG cells. We did the same exercise for the differentially expressed genes having a fold change higher than 2, and here we obtained the same results. So we can conclude that aflatoxin B1 is more potent than EMS in the human HepaRG cells. Of course, it's important to mention that the previous results were all based on the qPCR assay, which requires a high amount of cells and also is quite time consuming. And therefore, we also wanted to investigate whether we could increase the throughput of GENOMARK using next generation sequencing data. So as next generation sequencing is part of the genomic technologies that we use in toxicogenomics, which is a subdiscipline of toxicology to study the adverse effects on human health and the environment. I was looking into PubMed, for example, for the search query toxicogenomics, and here I found an amount of 2,200 results mentioning toxicogenomics in all fields in PubMed. And over the last 10 years, as we can also see in the trend of the articles published in PubMed, it's quite revolutionary that the amount of genomic technologies or type of genomic technologies is evolving very fast. So as from the discovery of the DNA helix, for example, when we started with DNA or RNA blotting or manually spotted arrays, these techniques are now replaced by higher throughput techniques such as RNA sequencing or targeted RNA sequencing that have a lower cost per sample since they require a smaller amount of input sample. And therefore, as a proof of concept study, we first investigated whether we could apply GENOMARK actually on gene expression data that was generated by RNA sequencing of an open data set. So in this article, a publication by the research group of Carol York, human HepaRG cells were exposed to 10 chemicals, 6 genotoxic and 4 non-genotoxic chemicals in 3 increasing concentrations for a period of 55 hours. So we applied the GENOMARK prediction models on the gene expression data. We found an overlap of 76 out of our 84 biomarker genes to apply the prediction models. And finally, we obtained the identical predictive accuracy of 90% for both models. So this suggests that GENOMARK is applicable also to data generated with other gene expression techniques. The results of the GENOMARK predictions on the different chemicals included in the open data set are depicted in the table here. And as I just mentioned, we had a prediction performance of 90%. However, 2 chemicals, cisplatin and colchicine, were classified differently by both models. And it was interesting that when we had a deeper look into the probability prediction scores for cisplatin by the random forest model, we saw a concentration-dependent increase in the predicted probability for genotoxicity. So when cisplatin was exposed in a higher concentration, we also received a higher predicted probability for genotoxicity. So maybe when cisplatin was exposed in a higher concentration, random forest would also have picked it up as genotoxic. However, the different classification results for both chemicals may also be due to the different exposure times, since in this publication, chemicals or HepaRG cells were exposed during 55 hours, while we use an exposure time of 72 hours. And we found an overlap of 76 out of the 84 genes to do the predictions. However, we had a high prediction performance of 90%. So this was a reason why we also wanted to continue and generate gene expression data ourselves by using the TempO-Seq. So TempO-Seq stands for a templated oligoassay with sequencing readout, and it is a targeted sequencing technique. And one of the main advantages of using TempO-Seq over RNA sequencing is that we don't need to perform the RNA extraction, since we can work directly with the cell lysates. So this decreases the time and cost that is required to assess a chemical. In the case studies, we used 2 different panels. We used one custom human GENOMARK panel. So this is the standard 1500 plus panel of BioSpider. However, here we also included all the GENOMARK genes, and therefore it's a custom panel. The second panel that we've used is the human whole genome or human whole transcriptome panel in collaboration with Health Canada and University of Ottawa. So for the study design, we seeded the human HepaRG cells, and after 7 days, the human HepaRG cells were exposed to 7 chemicals, 2 known non-genotoxic chemicals and 5 known genotoxic chemicals in 4 increasing concentrations versus solvent control for 72 hours. So at day 10, we collected the cell lysates, and the TempO-Seq analysis was performed by Bioclavis. And afterwards, we could perform the GENOMARK predictions on the gene expression data of the counts data. So in this table, we have the results of the GENOMARK predictions on the TempO-Seq data. So in the first column, we see the in vivo genotoxicity data for the different chemicals. And here we see the probability predictions or the output for the different concentrations of the 7 chemicals. So the first conclusion was that the 2 non-genotoxic chemicals were classified as non-genotoxic, except for eugenol in the highest concentration, we had an inconclusive result. And the 5 genotoxic chemicals were overall classified as genotoxic. And it was very interesting to see as well that we had, again, a concentration-dependent increase in the prediction score. So I did not show the individual prediction values, but when the chemical was exposed in a higher concentration, the probability prediction for genotoxicity was also increasing. And then for colchicine, which is an antigen, we had a not clear positive, but however, an inconclusive result in the 2 highest concentrations. Then we wanted to compare the prediction performance between GENOMARK and another transcriptomics-based biomarker for the hazard classification. So next to the TGx-DDI biomarker, which was a biomarker that is developed in a collaborative project between Hazy, Georgetown University, also the National Toxicology Program, and the Health Canada. So we used their online tool to upload our gene expression data. And the results of the predictions made by TGx-DDI are very similar to the output of the GENOMARK in the 4 concentrations. And this is very nice to see. However, so as well as for the genotoxicants, for example, we see that at the effect of genotoxicity is picked up at the same concentration. So however, for 2 chemicals, we see here something interesting. So colchicine, as I just mentioned, is the antigen, and by the TGx-DDI biomarker, it is classified overall as non-genotoxic, but in their case, it's non-DNA damage inducing. And in our case, it's predicted as inconclusive in the 2 highest concentrations. So here we see, for example, that both biomarkers can be used complementary, since TGx-DDI has another type of reference data set compared to GENOMARK. So, for example, antigens are in their biomarker are picked up in the reference data set as non-DNA damage inducing, and therefore it would be expected that it's classified as non-DNA damage inducing, while in GENOMARK antigens are in the reference data set as genotoxicants. And eugenol, as seen in the highest concentration in GENOMARK, is classified as inconclusive, but TGx-DDI is in all concentrations, eugenol is seen as non-genotoxic. So the main conclusion of this study is that both biomarkers can be used complementary in hazard classification. So what's next? We have used the TempO-Seq data, the panels, but we still have a lot of data. So we also want to perform the classification of GENOMARK and TGx-DDI for the data of the whole human transcriptome panel. We will perform a quantitative analysis on the TempO-Seq data by using the benchmark dose approach, compare GENOMARK and TGx-DDI for hazard calls and potency ranking. And finally, it's also interesting to mention that we are working for the validation of GENOMARK, and we submitted a pre-submission document to Earl ECFM, and we are at the moment waiting for their answers. So as a final conclusion, we can say that GENOMARK is based on changes in gene expression levels in human-derived metabolically cells, with the support vector machine and random forest-based prediction models allowed to classify compounds according to genotoxicity. We have some first indications for the utility and potency ranking of chemicals, and GENOMARK is applicable with higher throughput platforms such as RNA sequencing and TempO-Seq. And with this final slide, I want to thank my promoters of the VUB, Professor Van Haken and Professor Rogiers, also my promoter at Sciensano, Birgit Mertens, Stefan Verhulst, who is our bioinformatician at the university, and also the research group under supervision of Professor Carol York, together with the collaborators of Health Canada. Thank you for listening. If you have any question, I will be happy to answer you.